Morning, everyone. Can you hear me? Yeah, perfect. Okay. Rachel, are we okay with the Zoom webinar attendees? Yeah, all good. So good morning, everyone. Welcome for attending. My name is Morgan Morris, and we are just going to swap to our introduction. Okay, so thanks all for registering. We've got a really exciting lineup of speakers for you over the next few days. This is, of course, our tentatively first computational chemistry spring school. This is happening today, of course. This is our seminar series, and then on day 2, hopefully some of you are attending the workshops as well. So we have place for 60 people, I think, and they're all booked up. We have a really nice mix of biologists and chemists in both of the workshops. So just to give some context on how this emerged, I am a medicinal chemist, so I'm a final year finishing up PhD now with the University College Dublin School of Chemistry and Biorbirc as well. So Biorbirc is a Science Foundation Ireland bioeconomy research centre. Many of you will be from there, hopefully, from looking at the registrations. And we have a number of industry partners across Ireland, and that kind of involves things like forestry, agri-foods, and the marine sector. One of the things that I work with is lactose for targeted drug delivery systems. So we take lactose and we attach it to drugs. I'm a synthetic chemist through and through. One really positive experience I had in my PhD studies was I was seconded to the Cambridge in the UK by Crystal Cubed. It's a really amazing Horizons 2020 MSCA organization led by Professor Brendan Kennedy. He's here today and he'll be chairing one of the sessions for you. And this was to Cresset. Cresset is a computational chemistry provider in the UK. They do Comp Chem for the life sciences. So this is really the theme of our, I guess, seminars today, but also our workshops. How does one do Comp Chem in both chemistry, but also how do you apply these tools to structural biology as well and other fields that are less traditional in nature? So in my studies in Cresset, or in the research that I did, we did a lot of really exciting things with Comp Chem. I think I was talking to an academic in the School of Chemistry recently, Professor Susan Quinn, and she gave a really nice summary of what Comp Chem is and can do. So Comp Chem is something of a 5th pillar in the sciences, or the chemical sciences at least. We have organic synthesis, inorganic, nano material science. Then we have Comp Chem. Comp Chem is a different way of looking at molecules. So when you think of people working in a wet lab, you know, you make the compounds and then you go off and you might test them. If it's a biological assay, you might do asymmetric synthesis. That's for the chemists. And then for the biologists, you know, this is a tool you probably don't really use. Computational chemistry doesn't really sound like something you'd want to use in your day-to-day. So that diagram or video there was of me docking lactose into a crystal structure of a protein. And that was a bit of skills that I picked up from my time in Cresset. I work with zebrafish as a homology model for some of the stuff that I do. And that's something that I know biologists use as well. So, you know, you might use sequence data from UniProt, and you might want to put that into a PDB structure and get the different sequence and put that into the crystal structure for which you already have data available. And so we kind of looked at all of these different Comp Chem approaches and thought, well, this isn't really taught in UCD at all. In fact, we were looking at the registrations and having a bit of a giggle at the experience people came to us and said that they had. So it ranged from everything to nada, finito, nothing, sorry, I missed that one. So it's a really unmet skill set in our university, and maybe for some other universities as well, because I noticed that in our attendance list, we have some people coming from NUIG, we have people coming from Cork, several institutes of technology. So seems like it's something we could all do with a refresher course on, or see what it's about, and maybe it's useful for all of our research. When I finished, I went to the organizers, both of my PhD and also my secondment, so the School of Chemistry, Biorbirc, Crystal Cubed, and Cresset, and we kind of had a conversation about how we might turn this into school, maybe just to train people in on some of the methods and inform them what computational chemistry can actually do. I was also talking with Professor Marina Rubini, who's now our director of the MCCB, just there in the audience, and we don't actually have a medicinal chemistry module for Comp Chem and UCD. So this is something we kind of wanted to do and test it out on the postgrads and see if everyone liked it. It's a shame it's revision week. We could have done with some undergrads as well, but here we are, and we're all really excited to present what Comp Chem is and what it can do for your research. I just want to say a quick shout out to everyone who was on the organizing committee from these 4 organizations as well. From the School of Chemistry, Professor Rubini, obviously, my principal investigator, my supervisor, Paul Evans, also here today, managed the finances for this event, and also Marcus Baumann, who was involved in the venue management as well. Thank you very much to all of you. And of course, to all the people from ChemSoc, led by Aveen, for helping out with chaperoning people on the day. I want to also thank all the Biorbirc staff. So we had Professor Kevin O'Connor will be here for a different session on the Friday. He will be talking about industry engagement. Sadly not here today, but many of our support staff are. So Sean Douglas, Melanie, thank you very much for doing all the food and catering for today. I hope everyone has a nice time. It's all free. And Jessica as well has really helped in addition to Kathy. So thank you all for being here to help out. Crystal Cubed, obviously this event wouldn't be possible at all without Brendan's involvement and kindly providing me with the opportunity to go on secondment. So thank you very much for that, Brendan. And Yolanda as well, who's our project manager in Crystal Cubed. Maurizia Pendino, PhD student of Brendan's as well, really helped out with the event. So I also finally have to thank everyone from Cresset, before I introduce them formally, for coming here all the way from Cambridge in the UK today to talk to us in a very hectic schedule of talks over both today, but also tomorrow during our interactive workshops. What are our goals for today? Well, I suppose we have an unmet training need in computational approaches for the life sciences across UCD, but maybe some other universities as well. We want to extend this learning platform as well. So that's why we're offering it to online attendees also. We are happy to have, I think, 200 participants joining us over the course of the next 2 days in some capacity from, I think, 25 different countries. So we didn't expect that much interest. Maybe this is just a general thing that people don't learn Comp Chem. And then, of course, we wanted to make this free so everyone could actually join and there wouldn't be any registration costs involved that might prohibit people from attending. We wanted to provide a networking and research engagement opportunity for everyone. So we do have a poster session, of course, as well for everyone involved. Those of you who have come have brought your own posters. There'll be a poster prize competition later as well. And we'll be presenting these at, I think, 5:15, just before the wine reception. So we also wanted to try and tailor our content to the audience involved. So these workshops, especially that we're going to run on day 2 of this event, we've taken into account that a lot of the people attending from Biorbirc are a biology background, and so they won't be using these tools in a traditional sense. They may have different goals or ambitions for how to use sequence data or homology modeling for their own work and aren't particularly interested in the chemistry applications at all. So you're all very welcome, and we've tailored this course to you as well. Just to give a summary of our event over the next few days. So this is a 2-day training school, both today, the 27th, and then of course tomorrow we've got our workshops on the 28th of April. Day 1 is lecture style, so we're able to offer it as a seminar series and online as well through our webinar. And then on day 2, we've got our workshops, and I've just got some pictures there of the ALE rooms for tomorrow. So if they haven't been sent already, Rachel will be sending you details of how to get to the rooms tomorrow, well hidden as they are in the vast confines of the O'Reilly, or rather the Science Centre in general. Just a further word on the workshops as well. So we wanted to cater to as many people as possible, and so we offered 2 different tracks, and hopefully those of you who are attending the workshops are able to attend in some capacity. I think track 1 filled up in 3 days, 4 days. Track 2, we had a steady stream, but nevertheless that filled up quite quickly as well. We wanted to highlight as well the level of synergy there are between several of the techniques we're going to show over the next few days. So not just things like QSAR or homology modeling, but just to see how much overlap there is, surprisingly high levels of overlap between chemistry and biology using these tools in the life sciences. And that's why the first session for the first 1.5 hours is a joint one, where we're going to talk you through FLAIR as a package that's offered by Cresset. We'll have a break after that, and then afterwards, we will then split into 2 different streams. Those of you in track 1 are going to be introduced to Spark. That's a different software package offered by Cresset, which is involved in bioisosphere design. So take a small molecule, you want to design something similar that you think might have interesting biological properties, that's Spark. Then at the same time, concomitantly in track 2, we're going to talk through intermediate flare applications and how to manipulate protein structure. So at that point, we'll have diverged, but not by much. And following a lunch break, we'll go back then for the final session in the afternoon. Track 1 is going to go through an integrated workflow and how to use Spark with Flare. So Flare is kind of a general package offered by Cresset. It used to be Flare and Forge, but now it's just Flare. And it does a few different things that are of relevance to biologists, but also chemists. So everyone's heard of the traditional protein ligand docking in some capacity, but doesn't know much else beyond that. We want to kind of advance people's understanding across the next few days of what these are. I think the most crucial thing of all, though, is you're going to get a 60-day free trial to both the software packages when this is done. So if we train you in well, you might be able to use these tools for your own projects once this is ended. Just want to give a little pitch as well for our Computational Chemistry in the Life Sciences Academia Industry Forum. That's happening on Friday. It's from 9 to half 11 in the morning. So that's happening in E0.01. If you haven't registered, you can still do it through the Cresset website. That's a very interesting lineup of speakers as well. We have a number of industry partners attending, but it's completely relevant to PhDs and postdocs as well, or indeed academics. There is, of course, for those of you from industry, the opportunity to attend one-to-one sessions with Cresset on the day. But for those of you attending in a more informal capacity, we would like you to enjoy the panel discussion we have in the morning at 11. And that's with the director of Biorbirc, Professor Kevin O'Connor, Professor Brendan O'Kennedy, who's going to come in his capacity as the head of Crystal Cubed. And we also have Paulina joining us from the new AI Science Healthcare Hub in UCD. So it should be a really nice talk through some computational applications. And so that kind of leaves us then with the schedule for today. We have a number of different talks lined up. First and foremost, we have the welcome, the introduction, and then we have this general talk from the Chief Executive Officer of Cresset, Rob Scoffin, who I'll chair for the first session. Then we have a poster and networking session afterwards, so you'll get to see everyone's posters. We did ask everyone who did present a poster or has come with a poster to, in some way, just put a little twist at the end and give us a line or two on how they think Comp Chem can benefit their research. How do they think it can enhance what they do? And you don't have to know that. We just were interested to see the responses, both to see what people thought Comp Chem could do, and further to that, what we might be able to use the software for on their projects. Then from there, we have the afternoon session chaired by Brendan. He'll be talking through Crystal Cubed and the grant in place. And then after that, we have Scott will be talking from Cresset on proteins. So this is a little bit more relevant to those of you with a biology background, a little bit of structural biology here. I think Scott's going to be talking through things like PDB structures and several other aspects that may be of relevance. And then finally, we have Selma in that session. We'll be talking through a more medicinal chemistry angle. So how to generate new molecules for my research project. After the lunch, we come back at 2:10. So Colin will be dialing in, but before that, we have Kevin will be talking through how to predict how new molecules will behave. So, you know, molecular mechanics, dynamics, that kind of angle. And this will be chaired by Alison Reynolds, who is coming from Vet Sciences. And then we have a talk from Colin in Cresset, who is an experienced computational chemist. He'll be talking through a long career across GSK, and many others, including Cresset, obviously. We have another poster and networking session after that, food again, and we come back in the afternoon for 4 o'clock sitting, our final one, and Marina will be chairing that one in the afternoon. That'll be with our two probably quite interesting speakers from both University College London. That's our academic speaker, Edith Chan. She'll be talking through computational chemistry that she uses in her own research, so maybe of interest to several academics in attendance. And then we have Vicky's joining us from Globachem. So Globachem is a really interesting choice. They're an agri-foods company. And we thought just because we had a large number of people involved in the CDT shared between Biorbirc and the University of Nottingham, that we would offer Vicky or some company affiliated with agri-foods on the day and see how they apply their research in a marine or agri-foods sector capacity. And so she'll be talking about scaffold hopping and bioisosteric approaches as alternatives to macrocyclic picolinamides. And last but not least, we have the presentation of the poster prize in the afternoon. Marina, Marcus, and Rob will be going around with stern expressions in the afternoon and lots of boxes to tick on several forums. And then they'll be coming back to us at 5 o'clock to announce the winner of the poster prize. And following that, probably the most important part of the day is the drinks reception. So come along for 2 glasses of wine max, according to estates. On that note, I just want to say one last thing. So Alison is in charge of this, and she's here today. Hi, Alison. We have one survey we'd like you to do in the morning, and we have one short survey at the end of the day. So I think Rachel or Alison are sending you the link now, but we'd really appreciate it if the next few minutes you were to fill this in. So it gives us some idea of what you think of Comp Chem currently, what your thoughts are on what it can do. We're really interested to hear the responses, just to know your background as well. It's anonymized, so we're not going to know who you are. If you have a smartphone with you, we put QR codes outside on the reg desk, but it would be really nice if you fill that in over the next while. And then finally, at the end of the day, we have another short survey at about 5 o'clock, just before the poster prizes are announced. On that note, I'd just like to say thank you for everyone attending today. It's really nice to see everyone wanting to learn about Comp Chem and its applications across the life sciences, not just chemistry, but biology as well. I've had a really fun time coordinating this with our committee, and I hope that some of that enthusiasm rubs off today in some of the talks. It'll be really nice to see what people think of it, what people actually gain from the poster session, as well as the speakers. And it's always nice to have networking as well across different schools. We have people from SPBS, SPES, and the School of Chemistry, Vet Sciences, a lot of different colleges. I think 7 on the registration lists are attending today from across campus. And then, of course, those of you who are coming remotely, you're more than welcome online as well. And we also have a number of people who are attending from other colleges in UCD. So on that note, I'm about 4 minutes early, but if people wanted a few minutes to fill in the survey that we've sent out the link for, you can do that on your smartphones now, or you can do it on your laptops. We'd really appreciate that. And then I'll introduce the first speaker. Thanks very much. Okay, without further ado, I'd like to introduce our first speaker. So Dr. Robert Scoffin is an experienced entrepreneur and senior executive within the life sciences software industry. Following a DPhil gained from Oxford University, Rob worked for 8 years at Oxford Molecular, seeing the company grow from 15 to 250 people, and was involved in a successful flotation on the London Stock Exchange of the company. In 1998, he joined Cambridge Soft, where he was responsible for running the European team and oversaw the growth of the business unit to more than $5 million in annual turnover, and contributed to the development and growth in use of ELNs within pharma and biotech. Rob joined Cresset as CEO in 2010, and he now serves as the chairman of Cresset. During the time he has been leading the team, Cresset has further developed its software and contract research divisions from a very solid customer base into a high growth and profitable business. Rob also serves as co-chairman for Torque Software, a collaboration between Cresset and Elixir Software. So without further ado, Dr. Robert Scoffin can join us on stage, and he can share his presentation.
Thanks very much, Morgan. You can certainly tell when you've written your own bio. So it reads very nicely. Give me 2 seconds just to get my slides up. Okay, are those being shared all right, Rachel? Like that. Looking good? Super. Hopefully you can hear me if I wander around. I'm slightly mobile when talking. I feel constrained if I can't walk around. Thank you very much for the very kind introduction from Morgan, and thank you to Brendan and Alison and the rest of the team here at UCD for making us so welcome. Thank you to you for coming along and giving me a little bit of your time to talk about computational chemistry. And so when we started thinking, as Morgan said, about how we could kind of introduce computational chemistry, the thing that's fairly sort of daunting about that is such a huge field, and a field that's come along in an enormous way. And so what we decided to do was try and give a very high level overview across quite a broad area, but then apply it in specific areas. So you can see one aspect of how we actually go about applying computational chemistry. And the sort of natural place to do that is in drug discovery. For us, that's the predominant sort of customer base that we have for our software applications. But that's probably only 75 or 80% of our customers. So there's a very significant portion of the customer base who use the applications in other areas. So we will talk about that as well. So it's not exclusively about drug discovery, but we're using that as an exemplar to really kind of show how to translate from the theory. And there is some theory, I apologize early on because there is some theory, but how we translate from that theory into the practice and show how that actually can benefit in research across a very, very broad range of areas. Structurally, we've tried to join up the 4 talks that the Cresset folks are going to give. So myself, I'm going to sort of paint a picture and try and give you a very sort of high level overview. And then Scott will focus in on the sort of protein side of the equation. Selma's going to talk more about the chemistry side. So Scott talk more about the biology. Selma talk more about the chemistry. And then Kevin has the glorious job of tying that all back together again, sort of thinking about how small molecule and protein chemistry and biology, how those sort of come together. So one of the sort of first questions that we might want to get to is, what is it that we actually want to know? So what's the end result for all of this? And I always like to think about, where do I want to get to? And then I can sort of backtrack to figure out how to actually get there. So one of the things that has been a challenge over the last sort of 2.5, 3 years during the COVID pandemic has been thinking about how different sciences can get applied to rapidly develop treatments, preventatives, therapeutics for COVID. And you know, one of the things that it's a longer term sort of challenge is also questions like how do we treat particular types of cancer? So HER2 positive breast cancer or triple negative breast cancer, particularly bad condition, up until sort of 10, 15 years ago was fairly untreatable, but in the modern paradigm actually is treatable in a sense, as long as you catch it early enough, with lots of different methods of treatment. And so computational chemistry has sort of played a part in thinking about both of these. So I've got a little sort of example here, and so I've just stolen somebody's work, but I reference them afterwards. So here's a movie that somebody generated and posted up on YouTube of an animation of the spike proteins from COVID-19. And it's pretty and it's good eye candy, but actually there's really, really good science sort of sitting behind that, telling you about the dynamics of cell entry, so how that spike protein interacts with membranes. And so, it's a very complex calculation, but it gives you a lot of insights into what's going on. And similarly, we can do, so that's a reference if you want to sort of find it on YouTube. Similarly, if we're thinking about mechanisms, one of the mechanisms in HER2 positive breast cancer is an upregulation of a particular gene. So the ERBB receptor is overexpressed in certain cells, and that leads to proliferation, so overproliferation of cells. And there are a number of treatments that can be used to kind of combat that. So there's an antibody treatment, so trastuzumab, which can block the HER2 receptor itself, but there are also small molecule ligands. So lapatinib is shown down here. That probably easier if I do that. So lapatinib that can be used to interfere with the signal transduction from that particular receptor, and that can help to mitigate the worst effects of that particular type of cancer. And so these are great things to know. How do we actually get to know what this is all about? So I'm slightly a fraud in this audience, because my PhD is actually in inorganic chemistry, and I did a lot of physical inorganic chemistry for my PhD. So computational chemistry sort of came late in my development as a chemist. I did some computational work as part of my PhD, but then really got into kind of life sciences applications as part of the first job that I had when I joined Oxford Molecular. So I actually joined as a software developer, and then sort of worked my way through. So my natural tendency would be, well, we just want to measure it, right? So lots of things that we can find out about the physical world, we just go and measure it. Lots of things we can't find out about the physical world, so we can't measure everything. So the things we can't measure, well, let's model it. Oh, sorry, not that sort of modeling. How about, so this was the sort of modeling I did as an undergraduate in 1983. I started as an undergraduate chemist. Molecular modeling was that, plastic balls and springs and go and make a model of this thing. Not that sort of modeling either. So what we really want to be able to do is get to a physically real, but yet still interpretable picture. Pictures are great, but sitting behind that picture is a model that you can use to calculate properties of the system. In this case, so that is a kinase with an inhibitor bound to the active site of the kinase. So not ERBB, lapatinib, but a very closely related kinase and inhibitor. So that's the kind of use that I want to get to. But there are still things that are useful to measure, and we'll come back to what the measurements are going to get used for. There's a lot of sort of measurable quantities. So, you know, there's the chemists in the audience, and you're all mixed in, I guess. I bet you haven't. I bet all the biologists sat on one side, all the chemists on the other. That's what we would have done in my day. I'm sure you're all familiar with NMR. You know, if you want to know what's the structure of a molecule, run a mass spec, run a LCMS, GCMS, run an NMR, and then go and sit and sort of work out which of the protons, and you can elucidate the structure. If you want to know about the energy of a molecule, you can do some detailed calorimetry. So if you want to know the heat of formation of a molecule, if you're careful in your calorimetry, you can kind of work out heat of formation. If you want to know how molecules change over time, or how molecules change if you change the environment, you can do things like differential scanning calorimetry. So heat the sample up and look at the energy required for per unit of change in temperature. And you can look at phase changes, you can look at melting and freezing. You can look at many quite subtle processes. And those, you know, get applied these days in very, very subtle ways in protein structure. There's some amazing work gets done in the sort of DSC field. That was one of my techniques. So I did do NMR, but none of my NMRs looked like that. Those peaks were a bit sharp for what I used to do. I did do a lot of that, and my peaks looked exactly like that. Big, broad, nasty, sort of horrible stuff that I did in organic chemistry. Or you can look at effects. And so here's an example of a biological assay showing the effect of increasing concentration of compound and showing the biological readout. And that gives you a handle on the performance, in this case, of a single molecule. How is it affecting a particular biological system? So all of these are measurable quantities. That says, what can't we measure in brackets so easily? So there are things that, you know, we can't actually measure quite so straightforwardly. And one of the key ones is dynamic behavior. So understanding how complex biological systems behave over time is actually quite hard. So there are things you can measure about dynamic behavior, but it's pretty hard to get a lot of accurate information. And there's, you know, the quantity of data that you need is quite high. It's also very hard to measure small energy changes. A lot of physical measures don't give you the ability to go down to really small energy changes. And that has a particular sort of nuance, and one that we're going to come back to is really important. We're going to talk about through the day, is how do you separate out the individual components, particularly where those individual components are components of a large energy. So I have a rather trite example, but it's one that was used on me many times when I started getting into thinking about how to model biological systems. The founder of Cresset, Andy Venter, used to use this example all the time of how to explain the sort of difficulties inherent in understanding sort of protein ligand complexes. So if you pose the sort of theoretical question, here's the ship's captain, how much does the ship's captain weigh? Okay, and the sensible people in the audience will sort of say, well, you put the ship's captain on a set of scales and that's how you know how much the ship's captain weighs, right? But oftentimes in the sort of biological sciences, you don't have the luxury of weighing the ship's captain directly. And so the only thing you can actually do is you can take the ship's captain and you can put him on a boat. And then you can say, well, if I take the ship's captain and the boat, and then I take away the boat, what's left is the ship's captain, right? And so you've got a thing that weighs 1,000,000 tons plus a ship's captain, and then a thing that weighs 1,000,000 tons, and take the difference, and there's the ship's captain. And of course, whilst that is true, the difference between those two things genuinely is the weight of the ship's captain. Of course, the error bars on the two measurements is such that it's impossible to actually do that. So when you're thinking about the effect that small molecules can have on proteins, which we'll come back to as our example, which Kevin will talk about quite extensively in terms of how you sort of predict that. Bearing that thing in mind all the time. Proteins have a lot of energy, small molecules don't, but the subtle changes in energy drive a lot of processes in the biological space. So if we're not going to be able to measure that, or not straightforwardly measure that, begs the question, can we model that instead? And of course the answer is sure. So to any of these questions, of course we can. Then the obvious next question is, yeah, great, but how? How are we actually going to go ahead and model that? And the most fundamental way of doing that would be, well, quantum mechanics. It's physics-based, it's double quotes real. It's pretty hard. And I'll sort of come on to where that hard challenge comes from in the next slide. Or we could take a more sort of approximate approach. We can use something called classical mechanics. And again, it's physics-based, but it's an approximation. But it's fitted to physically real data. So that's where that kind of measurement stuff comes back. So we want to measure those properties of the system, because when we come to calculate, if we can calibrate our modeling based on physically real parameters, then we stand a chance of being able to predict what happens when I model something I haven't actually made yet. So that's a great thing if we can sort of fit that to observations. And I characterize it as slightly less hard, because it's still like a hard challenge to get it right. Mostly there are times when that's like really difficult. And then I'll sort of bring it back up to date with kind of AI methods after I talked about these two, and show a very specific example, a couple of very specific examples of how machine learning and artificial intelligence methods apply really nicely to this kind of physical modeling. So in the QM world, it's really easy at one level. I said it was hard, it's really not, because all you have to do is solve that. So there's a fundamental equation that governs all of this, which is the Schrodinger equation, and that's the time-dependent Schrodinger equation. So if you solve that, then you can get a picture like this, where you can look at an electron distribution in this case, so single electron in a box. Where's the electron over time? And that gives you the ability to then say, you know, both the static side of the equation, which is the bit on the right-hand side, so that's the density squared, or the actual wave function itself, and showing as that sort of varies in time. And then you can then also think about sort of snapshots. So you can take a stable state. So there's the time-independent version of that. So I'm sure you'll come across H psi equals E psi. So if you have the right formulation for the wave function, you can extract the energies from using a Hamiltonian. All sounds great. And if you do that, you get some pretty pictures. So that's what electrons look like. Job done. Albeit, of course, it's not, because it's fine for a single electron in a box. It sort of gets increasingly hard thereafter. Now, the good news is people have been working on this for 100 years now. So from the early 1900s, there are lots of formulations now that allow us to make approximations, and computer power has scaled to such an extent that a lot of this that 20 years ago we'd have been saying, well, you can't do that. Today you can take quantum mechanics and apply it to proteins. And it's feasible. It's just incredibly slow. And so for a lot of practical applications in an applied sense, it's just too long-winded. Not that it's, you know, sometimes it's too hard. Sometimes there are things you just can't do. But other times it just takes too long to use that method. So whilst that's great, we'd really like to be able to do something that's a bit quicker. And the classical approach to that is to use classical mechanics, and classical mechanics formulates chemistry to be balls and springs. That's it. That's classical mechanics. So atoms are hard spheres. They are joined together by springs, and then you have to have parameters to calculate how strong the bonds are. So what's the elasticity of the spring? So how far apart atoms like to sit. So you can get bond lengths, and then you parameterize the bending. So you get bond angles, and you parameterize torsions between four atoms. So you get the twisting. And if you pour all of that into a calculation, and you sort of separate out, so you can separate out the energies. So typically it's done, as you can see on the right-hand side there, typically it's done as the total energy is a sort of bonded term, so the springs, and then the through space, so van der Waals and electrostatics. And if you do those calculations, you can generate, let's say you want a heater formation, you know, what's the energy of that particular conformation of that particular molecule. So that's all feasible, and that allows you to get to the nice sort of 3D representations of complex structures. It also allows you to get to nice pictures of proteins and active sites within proteins. So we're getting there. And some of the sort of the benefits of molecular mechanics or force fields. So you'll hear us talk about force fields quite a lot. So a force field is just another way of saying what is the set of parameters that go into calculating energies and behaviors of molecules. Generally speaking, they are quick, particularly these days, you know, with compute power has sort of grown to such an extent that you can do a mechanics calculate the energy of a conformation of a, you know, even of a protein in, you know, sub-second on a CPU. So even on, you know, sort of laptop, you can kind of do the very, very complex calculations in quick time. Generally speaking, really good at reproducing structures and conformations, and that's because that's the data that was fitted. So when you're, if you've done your calorimetry carefully, you've got heats of formation of known molecules. If you do careful spectroscopy, you know from infrared, you know what the vibrational frequencies are, so you can kind of pour those in. These days, actually what most people do is just do complex QM calculations. So you just run a big density functional theory calculation to get the energy of a molecule, and then use that DFT as a pseudo for actually measuring the real thing, and you plug that into the mechanics. So you build these quite complex calculation engines where they tend to fall down. They're not scalable to unusual atoms. So if you haven't plugged in data for unusual atoms, then it's guesswork. So they tend not to be brilliant at things where you haven't parameterized them very well. And unusual structures where you've got some kind of second order or third order effect that's driving a structure, then again, if you haven't got data on that, the mechanics force field is just going to guess. And so it's not really going to give you a good answer. Where a QM calculation, quantum mechanics calculation, would probably get a better answer, certainly if you do a high level of theory, so do an ab initio. And one of the sort of bugbears of mechanics, if you're in the biological world, you'll have come across a force field called amber or charm. Those are sort of two that get used a lot. One of the challenges with amber and charm is that they really don't do electrostatics. So when I talked about the total energy in the previous slide in the non-bonded terms, one of the key things there is the electrostatic interactions, and they have sort of pseudo terms in their force field for handling those, but it's not very subtle, and so they really get kind of challenged by that. And this was where sort of Cresset came into being. So the company's 20 years old this year. It's our 20th birthday this year. But the technology that sits behind it, which we call the XED, or Extended Electron Distribution Force Field, that's been in development for more like 35 years. So this came out of work that Andy Venter did in the early days working with Sir James Black, who was a Nobel Prize winner and inventor of 2 blockbuster drugs in his career. So very, very sort of storied medicinal chemist, or he's a pharmacologist. No, you'd not be happy I had called him a medicinal chemist. He was a pharmacologist and he was proud of it. And when Andy started working with Jim back in the 1970s, the challenge was to understand why artificial molecules, synthetic molecules, behaved the same as natural substrates. So they were looking at phosphorus diesterase inhibitors, PDE inhibitors. And there was a series of sort of synthetic analogues that showed activity in various of the PDEs, PDE3 in particular, they were looking at, and they wanted to know why that was. So what was it about those molecules that made them similar to the natural substrate? And Andy, over a period of years, sort of came up with a formulation to explain not just shape, conformation, but also the electrostatics. And the way he did this was to model using extra atoms. So he inserted into the force field these, technically they're monopoles, so they have a charge, they have a single charge, they can be negative. You don't have to balance them out, so it's not a dipole. But in order to model the electrostatics, you sort of separate out the sort of nuclear component, the sigma part, so the bonded part of a sigma bond, a single bond, from the pi part. So in a double bond, you'd have some components of sort of pi orbitals, and using those dummy atoms, and they actually can get quite complicated. So here's a carbonyl, and you can see the charges are separated out, and the oxygen in the carbonyl gets actually 5 dummy atoms. And those all have parameters. So they have bonds, and their bonds have strength. They have angle bending parameters, and they have torsion parameters, which means they can move. And so this sort of gives you the ability to then think about what happens in a complex molecule as the electrons interact with each other. The charges on the individual atoms interact with each other. So it gives you polarizability. So the charge can move just as the atoms can move. And so this gives you a much more detailed ability to talk about the electrostatics. It gives you a much more physically real representation. And it allows you to do really good sort of structural minimization, energy minimization, and conformational generation. It's particularly parameterized and validated against molecules extracted from protein structures, so ligands of proteins, biologically active molecules. So that's our focus. So that's the data set that we use when we're doing that calibration work on the force field. We actually use large drug-like molecules because that's what we want to be able to reproduce. So it's particularly good at that. But it's a single force field that covers everything. So you can do Z calculations on just a box of water and it works really well, or a small molecule, or a protein. But the note at the bottom, I'll do that sort of for everybody who's online as well. The note at the bottom is don't think about doing this in dynamics, because there's 2 problems. One is you've got like 5 times as many atoms now, and so the thing is really slow if you want to do a dynamics calculation. But that's solvable, right? So that just throw bigger computers at it and you can make that one go away. Actually, the biggest problem is the dummy atoms have no mass. And so if you think about from a momentum perspective, if you put energy in, the thing that has the lowest mass will have the highest velocity. And so the thing that has no mass has all the velocity and therefore all the energy. And so what happens as soon as you put any energy into the system to do a dynamics calculation, all the Z's fly off. And they literally fly off. They just disappear into space. They do get very fast, very quick, and then disappear. And so that doesn't work. So when we come to thinking about the dynamic behaviors, which we'll talk about quite a bit, we're actually not using that force field. We're using a different force field, which I'll talk about. But what can we actually do now we've got the ability to calculate those kind of properties, well, we can do things like ligand similarity, so, or structural similarity, if you like. How similar are two molecules to one another? And that's that key foundational question, which was, why does this molecule behave the same as this molecule in a biological system when they've got very different structures? So they're sort of biosimilar, to use a phrase from the biologics world, but they had all bioisosteres is the sort of pharmacological medicinal chemistry way of talking about that. Well, if we can do that, if we can compare two molecules and say, you know, how similar are they and put a number on that, I can also search databases. So if I've got a database of all the world's chemistry, and I can take a single molecule and I can search against that. So in-house, we have a database of 20 million structures that you can, you know, compounds you can go and buy. And we can just do a search, take an individual compound and say, find me the 1,000 molecules that are most like this, that then I might want to test in whatever context it is. It doesn't have to be drug screening, it could be some other aspect of chemical performance, but find me the molecules that look most like this. We've done that for people in project work to find compounds which smell the same. We've found compounds which taste the same. So it doesn't have to be a therapeutic drug. It could, there's lots of places that chemistry gets used, and this ability to compare two molecules to one another and put a number on it is really powerful. We can also do protein electrostatics. So you don't just apply the calculation to small molecules, we can put that on big molecules as well. And we can also think about how, in the context of that sort of ligand binding challenge that is the overarching thing that we're talking about, how complementary is a ligand to a protein? And I'll show you an example of that. Morgan mentioned docking, so most people are familiar with the concept, or at least the word. Oh, we just dock a compound into that. That's great. But that's, yeah, generating an idea of how small molecules fit into proteins, or how two proteins would aggregate, protein-protein docking. Yeah, that's really powerful. And that's sort of the same force field allows you to do that. And then if you use a dynamics capable force field, you can actually go for what is currently kind of the state of the art, which is to generate a predicted binding energy. So rather than doing a expensive biological assessment of several hundred molecules, you can go and do the calculation beforehand and then make a smaller set of molecules to test your ideas. And again, we'll sort of come back to that a lot. What does that actually look like in practice? So if we take a series of substituted methylbenzenes, so here's if you can't see, that's fluoro, so difluoro and dichloro, and we run the Z calculation, and you can see that you get the electrostatics here denoted as a surface. The red is positive, the blue is negative, and so in the unsubstituted case, you've got this sort of slightly distorted by the methyl, but generally speaking, you've got this kind of classic hamburger of a sandwich of negative on the top and positive on the sides. When you start to substitute, the electronegativity of the fluorine atom pulls the electron distribution. So in the case of difluoromethylbenzene, it's almost completely flipped around. So that instead of having a sandwich, you've actually just got a dipole. So it's positive on one side, it's negative on the other side. And you'll see that if you look at protein crystal structures, as you start to substitute different atoms in, then the behavior will change. And it's the electrostatics that are changing as you make that substitution. And then you even get subtle effects. So in the case of the dichloro, the chlorine has a hole in the negative, and you can actually get a hydrogen bond. So in a protein structure, you'll sometimes see, if that was bromine, it's really obvious. So bromine is positive there. And so bromine to hydrogen bonds work really, really well. But you'll see this in the case of chlorinated phenols in a crystal structure. You'll see them making close contacts with the hydrogen, and it really is people call it a halogen bond. It's a hydrogen bond. by any other name, but you can get those calculations. And you can also look at sort of subtle, so I say, you know, take a calculation and look at a similarity across a series of molecules. This is the output from Spark that Morgan mentioned. So here we've started with a single structure on the left, and we've replaced a portion of the chemistry from a database of fragments. And so as you go through molecule by molecule, we're proposing different substitutions of the molecule and looking at, in this case, the change of negative electron density. And what you can do visually, as well as numerically, is you can see, you know, where the shape of the electrostatics is similar or where it's changing. So you get variation, but you've also got strong similarity. I like this example because this is Viagra. And if you take, so Viagra sells $4 billion a year for Pfizer. If you take Viagra and you put it into Spark, the second result that comes out is Cialis, which sells about $3.5 billion a year. And so if Pfizer had patented that single rotation of nitrogen atoms that have stopped $3.5 billion of competition in the market. The one on the far right hand side is also interesting, is that's not patented. So if anybody wants to get into the drug market, that was not covered by either Pfizer or the Biogen's original Cialis patent. I suspect it probably is patented now because there's been a lot more compounds have sort of been developed since then. But that's an interesting, you know, sort of simple 5-minute calculation that could have saved several $billion. I mentioned talking about the complementarity between the proteins and ligands, and this sort of dives into quite a detailed use of the electrostatics. So here I have 2 pictures, which is quite challenging. You have to kind of turn your brain a bit slightly inside out to work this one out. When a small molecule binds to a protein, a lot of that binding is driven by the shape. So the shape complementarity is important. Could be 75%, could be 80% of the complementarity is actually just driven by the shape of the molecule. But the remaining 30%, 25% is driven by the electrostatics on average. In some cases where it's really, really driven by the electrostatics. But what you really want to understand is where the molecule is positive, the protein should be negative, and vice versa. So the complementarity has to be inverted. So I've put a surface on the ligand on the right, I put a surface on the protein on the left and colored them by electrostatics. And what we're looking at is, can we tell where red is on blue and blue is on red, because that's good. But red on red and blue on blue is bad. So if you've all turned your brains inside out trying to work that one out, then in the software you can just do this, which is just recolor it so that where red is on red, it goes red, and blue on red is green, and vice versa. So green is good, red is bad. And so you can see here that the molecule is fitting into a cavity in the protein, and it's mostly good. But as a chemist, you might sort of be spotting this piece and saying, well, are there changes I could make to my molecule that would mean that it would actually make a more favorable interaction between the ligand and the protein in this portion of the molecule and sort of eliminate this clash. So using this kind of, I talked about, you know, pictures are really valuable. So pictures tell you quite a lot. In this case, you also get a number, so you can actually say, well, how good is that, or how bad is that? And that helps you in sort of progressing your thoughts and ideas and prioritizing. And one of the things I would like to say to people is, you know, bridge engineers gave up building bridges by building a bridge and seeing it fell down. Like, although the Romans stopped doing that. The Romans' approach was just build the bridge 5 times stronger than it needs to be. So that's why there are still Roman bridges standing today, you know, 2,000 years later. You just, yeah, over-engineer it. The Victorians had a slightly different approach, which is, you know, build the bridge down to be efficient, but then occasionally they fell down. And they usually fell down when it was windy or you put too many trains across them. And they hadn't really thought about that. And so when people started coming up with calculations to model the stresses and strains within bridge structures, engineers stopped just trial and error because it's so expensive. As chemists and biologists, we still, large part of the time, we still do that. We still say, I wonder if this will work, so I'll just make a molecule and try it. Whereas computational chemistry is that kind of thoughtful approach of saying, how good is this? Let me prioritize, let me figure out what might not work first, and then really just do the best thing first. And it's not usually a replacement for wet science. So, oftentimes people will say, one day all drugs will be discovered using computers alone. That's just rubbish, right? That's not true. But computers do help you significantly in making these kind of assessments. I talked about the sort of AI world, and that's a massive leap forwards that's happening across all the sciences. And sort of deep learning neural nets, the kind of stuff that Google have developed around AlphaFold, the ability to predict the fold of a protein just from sequence. But it also gets applied in the chemistry world, some really interesting challenges. One of them is really speeding up the very complex calculations. So I talked about how to build a molecular mechanics force field by doing DFT calculations, density functional theory, quantum mechanics. And on the right-hand side, there's an example there of a system called ANI that we're using actually as part of our force field parameterization. So this is how we use ANI at the moment. So whereas we would in the past have done extensive quantum mechanics calculations, and the example here is a set of molecules that each molecule would take sort of 52 hours on a single core on a computer. If you train a neural network to be able to reproduce the energy from a set of structures where you have done those calculations in the past, any new molecule takes less than a third of it, it's about a third of a second on a fairly standard GPU. So GPU that you have in your gaming PC at home can do 3 molecules a second versus 1 molecule every 2.5, just over 2 days. So AI, if you hear sort of AI, AI drug discovery is kind of a big buzzword. There was even an article about it in the Financial Times in London, which tells me that it's over, because whenever the Financial Times start talking about science, the science is done. But it's really useful in very specific areas. You can also do it with electrostatic surfaces. So we started doing some work with a group up in Newcastle in the UK, who've trained a deep learning neural net to reproduce electrostatic surfaces of molecules just from the structure, from the atomic connectivity. And that's amazingly fast. So again, it's sort of milliseconds of compute time rather than seconds for molecular mechanics or hours for QM. So it makes a huge, huge difference. And the sort of final thing, which sort of leans heavily into what the other guys are going to talk about, is really thinking about how you calculate the sort of binding energies of ligands to proteins. We always sort of focus in on Gibbs free energy, so delta G's, and the reason for that. So delta G gives you a measure of the work that's available for the system, the energy that's available in the system to do work. So what's the energy that's there to drive change? So in this case, we're sort of really thinking about change. And the change we're thinking about here is modifying the behavior of that big protein through binding a small molecule ligand to it. So the classic ship's captain problem. If we could measure the free energies of the protein and the ligand, then the difference between the two, and then we measure the Gibbs free energy of the complex. The difference is the binding energy. Yeah, it turns out we can't really do that. But what we can do is sort of extract that information from a molecular dynamics run. So in this case, we're going to use not the Z force field, but a force field that gives you the ability to calculate motion change through various time steps. And then that's going to give us the ability to simulate what actually happens in the real world. And then we're going to extract information from that using statistical mechanics. We're going to extract the energies for individual processes. So we might be interested in this case on the right, they're looking at how much individual residues move as they've gone through a series of calculations over nanoseconds. So 20 nanoseconds of simulation, moving a protein, and then looking at which parts of the protein actually move. And so you can identify, you know, in a protein is a little molecular machine, it's doing some work. In this case, there's a hinge and the hinge is moving. And so these residues in here are some of the hinge residues, which are showing the most motion in the simulation. So you're able to identify within the protein what are the key residues that we can then focus in on and look at. So if you wanted to stop that little machine from doing its job, maybe you want to sort of gum up the works, you want to stop that hinge from opening, then, you know, sort of something that could bind into a region that would affect those residues would be useful.
We're just at 7 minutes past 11. You're up.
I'm done. So that's how we got to our picture of the spike proteins. That's just a really big version of that last calculation. So complicated calculation, including the membrane, including glycosylation of the spike proteins, and looking at the motion. You can see if you look very carefully, you can see on the inside of the membrane where the spikes penetrated through, you can see some motion there. And sort of giving ideas about how you might want to go about sort of stopping that from happening. And that calculation we're going to come on to in quite some detail in sort of Kevin's talk, but it is actually quite complex to go through sort of generating that binding energy. And it comes from doing a very complex sort of thermodynamic cycle. But this is the state of the art in terms of how to generate binding energies these days. So if you want to know what's the predicted effect of a given small molecule, do your bridge building in the computer before you actually go and sort of make something that falls down. This is absolutely the state of the art. So it's sort of what's called absolute binding free energy, based on lots of those dynamics calculations, and then sort of extracting small numbers. Millions of numbers come out for the trajectory, and then one number, which is the energy, comes at the end. And you can also use that sort of absolute binding free energy or free energy perturbation, FEP, to look at a variety of small molecules. So in this case, an academic group in India were looking to find inhibitors of non-small cell lung cancer, so EGFR inhibitors, and they ended up making actually a very small handful of molecules from potentially thousands, and then coming up with a series, and there are crystal structures showing that those are bound to the protein and are active in inhibiting that sort of cell proliferation. So that was my, however high up helicopter view. Sorry if we covered a lot of ground, but that was what we intended to do, to hopefully sort of spark your thoughts and interests and ideas. And I'm very, yeah, you can applaud. I'm very happy to take questions.
So just in the interest of time, we might take one from the audience, one from the online chat, if there's any that have come through, Rachel. Does anyone have any questions so far? Yeah.
I was just wondering about this Z force field. Do you have to choose one confirmation? There's no dynamics.
Yeah, that's a really great question. So the question was, with the Z force field, do you have to choose one conformation to do the calculations and know. So Z also includes the ability to do a conformation hunt, as it's called. So you input a 2D molecule, and then it will generate a series of low energy conformers. And then for most of the methods that I was showing, you would use a population of conformations, so that you have a standard chance of having at least one of them that's close to the actual, the actual conformation that's active. Yeah, good question.
Anyone else? Yeah. Yeah.
Do you think that the Biorbirc plastic models from the 80s and 90s may still be useful at some extent in modern computation?
So do I think that the balls and spring plastic models from the 1980s are still used? I still have mine. I almost brought it with me. It was in the wardrobe at home. I almost brought it out. Yeah, in a sense, because I think there's a tactility to them that enables you to think about structure in a way that can sometimes be a little distant when you're looking at things on the computer. So things like stereo displays, sort of the ability to really try and see the three-dimensional nature of structures, that can help. I've done some work with VR headsets. It sort of gives me a migraine. I can't really do it for very long, but people who don't suffer from migraine, VR headsets can really help. So you can really kind of feel like you're interacting with the structure. But I still do like to think physically and the tactile nature of being able to look at the molecule, at the model, and sort of move it around sometimes really helps. But yeah, generally speaking, I tend to go to the computer first.
Okay, in the interest of time, I might just leave it there, if that's okay with everyone. We have our first poster and networking session now outside. We have our judges going around to look at the posters, and of course, that's an opportunity for some free food as well. Who doesn't like that? So I'll see you again. I think it's 10 to 12, we're back, and we have the next chair. Brendan will be on the stage, and we'll have our two speakers from Cresset. That's Scott, I think, and Selma as well. See you later.
Thank you.
Okay.
And start the video as well.
Do I need to start video?
Yeah, so they can see you online.
Okay.
Okay. Perfect.
Right. Okay. So I'm going to chair this session, which I'm very grateful to the organizers for. I wanted to thank everybody who's been involved in it. So Cresset and Biorbirc and the School of Chemistry. For my own team, Marcia, wherever she is, she's at the back taking photographs. Pandina, who's a PhD student, and then Yolanda Alvarez, who's in Spain, who have done the heavy lifting from our side and have contributed to it. But I actually really want to thank Morgan because he has, as far as I understand, chemistry being the catalyst for the whole thing today. Rob and I have talked about doing something like this and talked about it, but you actually made it happen. And it's great to see the people here coming together, the different disciplines. If any of the biologists were scared of some of the equations that Rob showed earlier on, we have included computational chemistry in our research, and I still don't know how to solve Schrodinger's equation. I still don't put atoms together. I still don't know how to program. I still don't know how to build a computer from a piece of string. Okay, so it's how far you want to go into these things. And I think the thing for us was, it was the goodwill of Cresset. It wasn't that they said, oh, you don't understand this. It was, we can work together. We can come across disciplines. And actually, Morgan's a perfect example of that as well, because he was never afraid to come over and try some experiments with the zebrafish, for example, or to go on secondments. So I'm going to give you a little bit about Horizon 2020 programs and RISE. Okay, I've only got 4 or 5 slides, and CRYSTAL3, CRYSTAL3 or Crystal Cubed, that's been mentioned, because I guess this wouldn't have happened without the fact that Morgan went on secondments to Cresset. So briefly, no, I'm going to do this one down. Okay, we are part of a RISE grant. Now, this RISE grant is to the whole of UCD, but it's also Trinity College Dublin are part of it, and so are Cresset. And in terms of all the different programs that the Marie Curie Actions have, RISE is about exchanging staff. So exchanging staff, so you go on secondment either from industry to academia or academia to industry. And in a newer version, some people can go from academia to academia. But the one we have is currently industry to academia, or the other way around. And you have to travel to a different country. Okay, so it's exchange of staff involved in research. And ideally, it's between different disciplines. So for example, we had forest engineers come to the School of Business to learn about machine learning, something you would never have thought about putting together. So it's, again, completely different disciplines, international networks who want from academic and non-academic is the technical term instead of industry. Okay, so anybody can go. Researchers, technical staff, admin staff, managerial staff, anybody who is involved in research. It doesn't mean that they are actually in the lab doing research. They can be coordinating research. They can be project managers of research, and it's about improving their skill sets. Why would anybody want to go? Well, the chance to go and develop your career, spending time in industry in a different country. We have lots of partners in Barcelona, for example. That's a great place to go and visit. But the other reason is there's a financial incentive as well to everybody. For each month that you go on secondment, you get a top up of €2,100 to your allowance. Okay, for a PhD student, that is very attractive, I'm sure. Depending on the sending institution, that can be completely regarded as mobility and is therefore tax-free. Okay, you have to go for at least a month, though it can be split up into shorter periods of time, but it has to amount to a month. And then whoever's hosting you will get $1,800 a month in the consumable budget. And then the rest is the 700, which is involved in overheads and managerial costs. So there is a real incentive here. There is a lot more criteria than this, okay? But all of these things we're able to overcome. So again, RISE grants in general, something that the academic and industry partners might think about being involved in a RISE grant, and then individual researchers, you might want to be involved in a specific one. The specific one we have is called Crystal Cube. This is our 3rd version of the grant. These are all of the partners. So you can see Cresset is involved there, Trinity College Dublin, UCD, and again, it's the whole of UCD. It's not just my lab or not just the Economy Institute. It is everybody in UCD that can go on secondment as long as they are linked to research. Justine O'Brien, a postgrad in my lab. So for example, she probably can't, she's smarter than me, so she probably could solve Schrodinger's equation, but she went on secondment to Cresset at one stage and looked to identified better versions of molecules that we were interested in based on a computational screen. And so that was very useful. That has led to grants. It's led to her thesis. It'll lead to a publication soon. It led to an invention disclosure. For example, then she went to Experimentica in Finland and was testing those in vivo models afterwards. So it's a network of bringing people together that have different expertises. So we have industry and academic partners in here. You don't need to read this. The grant is focused on something called the cysteinoleucotriene pathway, and these were the receptors. These are G protein coupled receptors, and Morgan was looking at these structurally and docking molecules to them. But it's actually about a lot of diseases like blindness, central nervous system disease, cardiovascular disease, and cancer. These are very broad grants. So it's easy to bring people in from very different areas. Okay, the real part is probably down here. It involves computational discovery, biochemistry, pharmacology, genetics, engineering, optometry, preclinical disease modeling, marketing, and commercialization. So there's always an opportunity to fit someone in, even if you don't work on cysteinoleucotriene receptors. That's just a focal point. Oh, sorry, this is the older version. So these are our current Marie Curie fellows. Edward, Alex, Almedina are here. Morgan is here. I don't know if there's anybody else here. Justine is shown here as well. So if you're interested in this, contact myself or Yolanda. We can explain to you. We can look to partner you up at one of the industry partners. If you're coming from academia, if you're coming from industry, we can partner you up at one of the academic partners. And I think it's a great opportunity if that's something that you're interested in doing. So again, this is how we're here today. How did we end up here today? Because Morgan, I don't know where I met Morgan, but I somehow convinced him that it might be a good opportunity to go on secondment, and he was very much interested in it, went to Cresset, did a lot of great work in terms of our own research, but then has put this event together. So well done to you, Morgan. Okay, that is my sales pitch for Horizon 2020 and RISE and Crystal. And now we're going to start the next session. So I'm going to try and multitask here, which I probably don't have the right chromosomes for. But we are going to have a couple of talks now from employees of Cresset. So the first one is Scott. He completed his PhD in theoretical materials chemistry at the University of Reading. Experience includes classical and quantum mechanical methods for stimulating excited states, thermodynamics of geochemical processes, and electronic properties of conductive materials. Okay, Scott joined Cresset's postdoctoral program directly from his PhD in 2021, has redirected his skills towards drug design, applying his knowledge to study the behavior of ligands and proteins. His broad computational modeling experience, as well as Python and machine learning expertise, allowed him to become involved in multiple areas of Cresset, including development of new software features and aiding Cresset Discovery consultants on research projects. Okay, so I'm going to welcome up Scott. And again, we'd love if you have questions at the end. We have, I think, about 25 minutes and then 5 minutes for questions. And if you're online, please post questions in the question and answer box. And I am trying to get you set up here. This is you.
Lovely.
Is that sharing the screen?
Not yet.
Because I got out of sharing the screen.
If you go back into presentation mode and then do the share. That's way to go.
I know you just told me that and I'm going to ignore you. Is that sharing the screen? Is that sharing the presentation now?
Looking at Rachel.
All good? And you want to hide that thing.
Lovely.
You're all set to go.
Thank you, Brendan. Hello, everyone. Right, I'm Scott. I'm from Cresset. So we had a really nice introduction to
computational chemistry this morning. I think Rob did a brilliant job of covering a whole computational chemistry course in about an hour. This talk's going to be slightly more focused. We're going to think about the kind of, what do we think about when we're trying to model proteins specifically? You're going to hear talks from Selma and Kev later, and that will be coming at computational chemical design from what we call a ligand-based perspective. So that's when we think about the actual molecule that we're going to change. One of the things that we need to think about before we can worry about any of that is the protein target. And that's basically going to be the topic of my talk today. And before I start, I'll apologize to the biologists in the audience. This is going to be a little bit basic towards the beginning, but we have a massively eclectic audience, which is really, really good. So I want everyone to kind of be on the same level before we start talking about some of the computational methods. I hope it's nice and accessible for everyone involved. Oh no, the clicker doesn't work. These are the buttons.
It probably will work if you give it a moment.
Oh, is it?
Click on it with the mouse.
Click it, yeah. There we go. Right. So what are proteins? Why do we care about proteins? So large biological macromolecules composed of amino acids. I'm sure that's not news to anyone in the audience. I'm really going to focus in on enzymes because they're a huge class of target for computational chemical design. Generally responsible for biological catalysis, and generally, very, very generally, they're there to speed up biochemical reactions. And because they tend to work in a single direction, you tend to have enzymes that work against each other. You have them in agonist-antagonist pairs. So the really simple example that I've given there on the screen is the kinase phosphatase enzyme pairing. In one direction, it adds a phosphate group onto ADP, and the other direction, it takes a phosphate group away from ATP. And this whole kind of push-pull motion, chemists call it equilibria, biologists call it homeostasis, but really it's just about maintaining order, and that's something that goes wrong from time to time in biological systems. So why do we care about enzymes? Well, when enzymes start misbehaving, that's when that can be a factor in leading to pathology. A really broad example is cancer. And cancer, the growth of cancer cells can be in some cases attributed to overactive kinase enzymes. And this beautiful diagram that I've drawn here shows you a big kinase arrow. The kinase is misbehaving, and we have some problem in the body that's not supposed to happen. So what can we do about that? The typical model of substrates and enzymes, which I'll try and get this in here. This I'm sure everyone's familiar with. It's the super simple lock and key model. Substrate, enzyme. Substrate is the thing that reacts at the protein. So how can we start to model the behavior of proteins? So Rob introduced what the idea of a model was. It's a computational representation of the problem at hand. And we do that so we can find some stuff out without having to measure it. So this is the sort of diagram that I'm sure everyone's really familiar with. What we generally call the lock and key model. So it just basically encapsulates the information that a substrate shape really determines whether or not it can fit into an enzyme, and enzymes are specifically tailored to accept specific substrates of a particular shape. So this is about as simple as it gets, but it encapsulates a lot of information, which is useful on some level. This orange diagram on the right is the simplification, and you will see that in almost all of my slides. So I hope you like it. What I'll be talking about is how we block the region, the triangular region in the enzyme, which we call the active site. And that's where the interesting chemistry happens. And if we want to modulate protein enzyme activity, we need to do something to that active site. So a slightly more advanced model, and again, it's not going to be news to very many people in the audience, but I'll go over it nonetheless, is a slightly more advanced picture of how proteins are formed. So on a very basic level, they're formed of amino acids that are formed of carboxamide linkers. This forms a primary structure, a 1D array of amino acids. Secondary structure, when we start to have some folding, beta sheets and alpha helices, we then have a tertiary structure, which is composed of our secondary structure formats. And at the tertiary structure, we really start to have a 3D thing where some interesting biochemistry can happen. And quaternary structure kind of gets an honorary mention, although for the purpose of this talk, it's not too important, but it's an accumulation of tertiary structures. So on the left, we have the diagram, which is basically our secondary structures. So we've formed a protein on secondary structures. This can be really, really informative. We can see where the very stable alpha helices and beta sheets are. Unfortunately, though, if we're going to do computational chemical design, we need to go further into the problem and we need to think about atomic structure. And as you can see from the diagram on the right, this is the same protein. It's an enormous molecule and it's really, really difficult to handle. So this is generally why, if you can avoid thinking about proteins in terms of atomic structure, then people generally do. But to design drugs, we really need to think about the atoms involved. So I mentioned the active site before, and that's where the biochemical process goes on. The active site is generally located in a region of the protein that we call the pocket. And if you think about the chemical structure of the protein active site, that's what the drug sees. There needs to be bits in the right place for the drug to be able to stick to, otherwise it's not going to work. So on the left, you have a diagram. This is from Cresset Tools Flare. So for those of you who are joining me in the track 2 session tomorrow, Flare has tools to be able to identify potential pockets and active sites in protein structures from a computational perspective. In the middle, you have an isosurface that we put onto the protein, and you can just see in there, this is the pocket, and this is a drug molecule that's blocking the active site. So this is now a protein that we're trying to give some therapeutic effect to. This can be for medicinal chemistry purposes, but it can also be agrochemical. This could be a pesticide or herbicide. It's just blocking the protein functionality. This is the region where the reaction normally happens. We now can't do that because we've put a small molecule into that region. That only works if that small molecule is happy in that region. And basically the computational approaches in this kind of field are to give you an idea of is this molecule a good fit or not. Back to the trusty diagram, we want to block the active site, as you can clearly see there. So a detailed look at the active site. Well, because this is where most of the interesting chemistry happens, we can kind of reduce the size of our problem to make it tractable for reasonable computers by just saying, well, we'll forget the broader structure of the protein. And basically what we'll do is we'll just look at the active site. And that is an approximation that holds in a lot of cases. Sometimes the wider protein is extremely important and you can't neglect it. But computational chemistry is kind of all about making the relevant approximations at the right time. So what we want to do is we want to build a structural model that looks like this, and we want to basically have what we call a ligand. This can be a drug, it can be any kind of agrochemical active compound, and what we want it to do is stick to the active site. So on a very, very basic level, the diagram on the right is something that we can start to do in software products like Flare, where we can look at the contacts between the ligand and the protein. And that can start to give us an idea of the chemical happiness of that relationship. So first kind of method that I'm going to talk to you about is something that Rob already touched upon, but we're going to talk about it from a kind of protein orientated perspective. Atomic arrangements in general cause electronic environments. You can model, if you put a sequence of atoms together, there will be an overall electronic isosurface, and that we kind of approximate, and we call that electrostatics. We call that electrostatic behavior. Overall, as Rob mentioned earlier, you can have regions of electrostatic positivity and negativity, and whether or not the electrostatics on the ligand line up with the protein is really, really important for understanding whether or not that ligand will bind to the protein. So the image on the right, you've already seen something similar. Green regions indicate that positive and negative regions are lining up nicely. There's good chemical attractiveness between the ligand and the protein. Regions in red are places where the chemists might want to adjust the ligand to make it a better fit for the active site. And you could kind of, that improving the ligand in that way could lead to essentially better properties. So this is how kind of on a very broad level, computational drug design can help inform the practical chemistry. So this is docking is something that I'm sure many people have heard of. The actual details of it are quite complex. It's something that we're going to look at in a lot of detail in the workshop tomorrow. But on a slightly broader level, docking is just used throughout the life sciences. It's a fantastic tool, and it basically it gives you information about the protein ligand interaction based on what we call a pseudo energy function. So Rob earlier described two main methods of calculating the energies of collections of atoms. You can have quantum mechanics or you can have classical mechanics. This, because it's dealing with huge proteins, generally uses classical mechanics. And what we do is we put the ligand into the protein, our ligand, and we give it a score. We say, what's the energy of the ligand in the protein at this orientation? We can do stuff like allowing the protein to move slightly, we can allow the ligand to move slightly, but generally, if we put the ligand into the protein like this, we give it a score based on its energy. And then what we do is we change the orientation of the ligand and we give it another score, and we work out the energy and so forth. So basically what you get out of docking is what we call scores. So a good score is related to a low energy pose, and in nature, low energy equals likely to be found. A bad score would be a high energy pose, and that's unlikely to be found in nature. And then we can, so we're not always restricted to just, well, in fact, we're very rarely restricted to a single computational method. We can do things like looking at these poses. So this is my super simple diagram of the triangular ligand in different poses in the active site of this protein. I think it's, well, if I was to look at this, I would kind of say, well, this one on the left, this is probably going to be the happiest. It's the best fit. It maximizes the contacts between the ligand and the protein. So this I would expect to give you a good score. This would be a good score in pose, as we would call it. And the pose is the orientation of the ligand. So a quick sidetrack about the energetics of proteins, which is really important to think about before we can start going on to ligand design. Generally, we use crystallography to look at protein structure. There are several, crystallography is kind of the best thing that we can do to try and take a snapshot of the protein. But there are several limitations to using crystallography. The most obvious one being that to get it to work, we have to have it in a solid state form, where of course if it's in the body, it's in solution. So the protein is in a completely different state of matter. So crystallography isn't the complete picture, I think is the take-home message. When we do computational chemical design, if we want to design the ligand, the small molecule, we generally assume that the protein is just fixed in a low energy kind of happy state. And as Rob mentioned earlier, we just, when we think about the internal energy of a molecule, that can determine its behavior. And we just think in this case, the protein is a big molecule, and we just want to find the lowest energy kind of states of the molecule. The biologically active form will be one of those low energy states, because high energy states aren't found in nature, certainly not in human bodies. So as Rob kind of touched on with his captain's ship analogy, the protein is huge compared to a small molecule. So the protein has to be happy, or you can't model it. It's not going to work. And I've got this in-depth diagram on the right that shows you an unhappy protein. Even if you have a fantastic ligand, brilliant drug molecule, it will not work. You will get rubbish when you try to model if the protein is not in a happy state. The protein has to be happy, and happy, energetically happy, found in nature. That's the mark of a good model, is whether or not we're representing nature well. So that was a kind of sidetrack just to say that protein energetics are super important, but we want to get to a point where we don't have to think about them anymore, that we can say, right, we've found a good state of the protein, now we can work on our computational chemical design. So let's assume we've taken a crystal structure and we say, no, there's something wrong with this. We have to manipulate the protein structure somehow, and computational methods allow us to do that. We avoid it where we can, because it's very difficult, but sometimes we have to kind of bite the bullet and say, okay, the protein's going to have to move. What can we do? Generally speaking, you need much larger computers to model proteins at the same level of theory as you need to model small molecules, because they are so very big. And we call that computational cost. That's something that works its way into computational chemistry kind of all the time. What's the computational cost? Now we can do things called protein minimization. Minimization is not a dynamic technique, but it does allow the atoms to move. And I know that can be very confusing. We do still call minimization a static method, even though the atoms move. And that's because the calculation is in principle performed at 0 Kelvin. So there's no external energy acting on the system. And what you will do is you will only ever get very small movements in the atoms in your system. By contrast, protein molecular dynamics is something that I'm sure a lot of people are kind of familiar with. There we allow kinetic energy to enter the system and leave the system. It allows you to do a whole lot more at the expense of computing time. So that's always the trade-off that we're making. We're saying, well, we can represent things better, but it's going to cost us more computing time, and it's going to take a lot longer. The good thing about protein MD is that we can put a temperature in. We can model things at kind of biological temperatures. We can allow a solvent to be present. However, if you look at this diagram on the right, this is a kinase enzyme in a box of water, and it's got 40,000 atoms. To put that into context, some of the systems that I was looking at for my PhD, where we were using UK's tier 1 supercomputing facility, I was kind of doing quantum mechanics on about 10 atoms. So we need a supercomputer. So this is an enormous collection of atoms to get a computer to think about. So just this is basically a comparison between the two methods. Optimization, you'll typically see very small structural changes. It can allow the energy to relax slightly, and it's relatively fast, but you aren't going to get any big changes. Molecular dynamics, however, much more going on. You can allow you're allowing things to move around according to Newtonian mechanics. And you can basically do a lot of analysis on these types of things, including free energy perturbation methods, which Rob introduced earlier. So moving away from actually manipulating our protein structure, what happens if we don't have a protein structure? What if we don't know actually what the protein looks like? How do we start? How do we start there? The first thing I'll kind of touch on is homology modelling, and I'm sure there's a lot of people in the audience who are far more familiar with homology modelling than I am. At least in principle. The use case here is where we have, for example, a piece of DNA that encodes a human protein, and we have a piece of DNA that encodes say a rat model protein. This is kind of key for the drug design process. Where we don't know how one of those proteins fold, but we can look for similarities between the protein primary structure, the DNA, we can use one as a template to the other. So what we do is we look up, we have a protein primary structure, we look for sequence similarities, and then we map regions of that onto a template where we know how the protein folds by crystallography or something like that. It's just overlapping and looking for template alignments. Very successful technique and something that Cresset use a lot. AlphaFold is probably slightly more cutting edge. Very, very new thing developed by Google DeepMind. Is extremely impressive. And you basically, all you have to do is put in a protein primary structure. You'll have some kind of AI technique that has learned how protein primary structures usually fold, and it will give you out a 3D structure of how it thinks it will fold. Now, this is artificial intelligence. It uses a type of neural network. It's very good, but it's not yet at the point where it can replace experimental data. So at the moment, kind of homology modeling, I think, is preferred where it can be used. So I'll wrap up there. It's basically just a kind of a very quick walkthrough, hopefully to get the audience members thinking about the kinds of things that we have to do in computational modeling. Proteins are essential in biological processes. They make a very good target when we're trying to make active chemical compounds. Drugging, for example, a particular protein target can lead to a therapeutic effect. The protein structure is therefore very important. Happy protein equals happy drug designer. We have to get the protein bit right first. That's because a small molecule cannot overpower the protein. The protein has to be deserve some special attention. We can manipulate the structure of the ligand or the protein, but generally where we can, we do ligand-based drug design, and we usually assume that the protein is fixed. It's a fixed point of reference, and then we can start designing our small molecules around a fixed happy protein. And this is kind of where I hand over to Selma in a few minutes, where she will kind of go over some of the more ligand-based aspects of this kind of work. So happy to take questions. Thank you very much for listening.
Okay, thank you very much, Scott. Is there any questions from the audience? Oh,
No.
young man there.
Scott,
Hiya.
fascinating talk. So I had a question about computational costs
Mhm.
in terms of the contribution of interactions as a driving factor for computational costs versus the number of atoms involved. So,
Right.
for example, if I wanted to do allosteric modulation of my ligand binding pocket, right, the protein was very well characterized, the ligand protein interaction was very well characterized, but now I was adding in an additional factor. That's not a lot of atoms, but it's a lot of interactions.
Yeah, so the what most computational methods boil down to is a multi-dimensional optimization problem. So you're adding in vectors in hyperspace for the optimization problem. So I don't know how allosteric modulation scales personally. That'd be something we can kind of talk about. Generally speaking, atoms will contribute to computational costs, but then you can, if you want to simplify your problem as a more general kind of answer, you can turn off specific interactions. You can modulate computational costs that way. So if you know that things aren't important, things can be ignored or they can be added in. Whether or not that's linear scaling is a question of individual is an individual question. But generally you can have atoms that don't interact, so they won't add that much to the computational cost. But yeah, in terms of your specific use case, I'm not entirely sure. So apologies for that, but great question.
Okay, any other questions, Jared?
Thanks for the talk. I was interested in that scoring system you're using with the small molecules fitting into those active pockets. And I just wonder, is there, you have a kind of a success rate?
This one. Or this one.
This pose.
Yeah.
Do you have a success rate whereby is there kind of a threshold score computationally that you would have confidence that when you go into the lab, it actually convert into a
right. That's
kind of relatively
so generally in computational chemistry, it's extremely difficult to get absolute values. So if we take if we want to compare, say, two completely different ligands, they're very difficult to compare. But you do get a score that can be affected by ligand size, flexibility, so forth. If you take two ligands that are the same, the different poses will kind of will tell you something. But it's the point of comparison basically is the issue. Generally, we look for a threshold score, as you're saying, where we're kind of happy or not, but it's very case specific. You have to be very careful what you compare it to, because those scores can be skewed hugely by external factors that you also have to think about. So in all of these things, you can never really put 100% trust in the number. It always has to come with a bit of kind of looking at it and saying, does this make sense? But I know what you mean. And when we do docking, we generally look for what we call we use a product called Lead Finder that's built into Flare. We usually look for a score of 13 or above, and that's telling you that this is really good now. So yes and no, I guess, is what I'm trying to say.
Maybe I'll add to that, because we had a slight conversation outside that might have been part of that. I think it also depends on the assay you're using. So if you are just looking for it to actually physically bind to the receptor or target, whereas we were adding the drugs into a zebrafish in a plate, so all of the pharmacokinetics could be issues about some of the drugs not being active. So yeah, I think it depends what your biological assay is at the end. Rob, yeah, it's right. Yeah, there's a question online.
Yes. Roughly, how do you correlate computational chemical happiness with actual binding and bioactivity for a given ligand protein interaction?
So yeah, so I guess the question of how do you yeah, how do you know if the protein's happy? Is what I'm kind of looking at that. As I didn't come from a biological background, that was actually something when I joined Cresset that was extremely difficult to pick up. I think your the biologist and chemist's brain when looking at a protein is ultimately the best tool, and that's been proven by Big Pharma several times. The sort of stuff you tend to look at is how well your protein is prepared, protonation states of the amino acid residues in the active site, amongst lots and lots of other things. It is a massively empirical thing to prepare a protein. As I mentioned, the biologically active form will be one of many kind of what we call low energy states. That means that there's not clashes between residues. Chemically, as with a chemical mind, you can say, well, that looks good, that doesn't. These dipoles aren't aligned. All of these considerations. What we want is to just define basically one of those happy configurations, and there will be many. And if you give a protein to two different kind of biochemists, they will probably prepare it differently. And that doesn't mean that one's right and one's wrong. There are just lots of right answers for proteins because they are so complex. So I hope that kind of answers the question to some extent.
Okay, I think we're in the interest of time again. We'll move on, and now that everybody's proteins are happy after that talk, we'll give that hand over to Selma. So thank you very much, Scott. Is that sharing online, Rachel?
I think it's sharing in that green.
Oh, that box. So Rob is going to walk me through this, right? So I am going to go stop share. And then we're going to go.
The other one. You can
Scott,
close
isn't
it
it?
down. Yes.
So we can close it down.
It should be
From beginning.
and then if you hit Windows key and go to Zoom and look in the taskbar at the blue icon at the bottom there.
And
Return to meeting with getting floating control now.
and it is.
Share screen.
Screen the top one. I think it's, yes.
All good? Perfect. Okay. So
How can you get a job doing this?
so Selma is a medicinal chemist with expertise in autoimmunity, CNS disorders, and pain targets, a track record of delivery from target to candidate selections, thrives working at the interface of computational sciences and medicinal chemistry, applying novel computation approaches for innovative compound design, which have led to multiple candidate deliveries. She's over 16 years drug discovery experience gained at Eli Lilly and holds a PhD in organic chemistry from the University of Bath. Selma is part of the Cresset Discovery team, providing medicinal computational solutions to pharma, biotech, and academia, and is working to streamline the drug discovery process with the integration of the TORX platform.
Yes.
Hopefully I didn't mispronounce any of that.
Thank you. Thanks. Thank you for the introduction. So, hi everyone. So the title of my talk is about how do I generate a new molecule for this medicinal chemistry project. Not really medicinal chemistry project. This could be applied to any molecular project, but I found it easier to talk about a medicinal chemistry project, so I will go through that. Okay. So the outline of the talk, I will touch upon the example of the drug discovery process, talk about the current challenges that we have, particularly in terms of management of the process map, but also in terms of how we can improve these processes. So I will go through the different hit generation strategies that we are using, and also introduce some in silico tools that we can use as a medicinal chemist without too much computational background and use those to really improve the rationale of your compound and hopefully impact impact your biological hypothesis. Okay. So this is a busy slide. I apologize. I just wanted to give you a brief overview of how the drug discovery process looks like. It is a very lengthy process, over 15 years in general, and up to here, it's what we call preclinical research, and then you have your clinical trial, regulatory approval, and where you have all those marketing and selling people coming in and talking to the physicians. So I'm focusing on this side of the discovery process, and I will, for today's talk, will cover mostly this region. So you can see in terms of number of compounds being made, synthesized, and number of compounds getting into the clinic, your probability of success is pretty low. So long process, pretty low probability of success. And then, so you want a return on investment. So you have patent for that. And the patent life is usually around 20 years. And you're not waiting for the compound to enter your clinical trial to patent a compound, because otherwise all too easy for the competition. So you will start a bit earlier and again eating on your timeline in terms of patent protection. So expensive process, a lot of iteration in terms of when you have your biological targets, how do you validate that? How do you prove that you have pharmacologically active compound, but also how do you leverage the off-target pharmacology, side effect, etc., but also think about the dosing, the therapeutic dosing that you need in order to manage those toxicity issues that you could have. Okay, so.
Sorry, just no, can you try and stay in the middle? We lose sound on Zoom and go to the far side.
Okay, so we need a more efficient process, and for this, so you can look at how 10,000 compounds is a lot of compound to synthesize. If you're the one in the lab, you will never see the end. So how can you really reduce the number of iterations to test a hypothesis? How can you do that? You need to also think about reducing the cycle time. And as I mentioned again, apologies repeating, reduce the number of iterations. So there are a lot of ways you can do that. And it's about looking at mitigating these issues by having better starting point. So thinking that if you have a starting point that is already selective, you might have less issues later on, but also thinking of you're going to start with the compounds, but you're going to, the more you will go through your process map and validate your biology, you will be optimizing. So you need to really leverage that. Okay, so what I'm going to cover now, I'm just going to talk about how I can generate new ideas for a new project, basically. Okay, so there are several strategies in terms of hit generation. So what we call hit is a compound very early on in the project where you're demonstrating some pharmacological effect. Most of the time it will be an in vitro activity, okay, and on the cell-based assay. So imagine you're a biologist, you have an interest into a particular target. There is nothing much in the literature, but you feel that with some genetic studies and etc., that there is a background that will tell you that if you either inhibit or activate that biological target, you might end up with having a therapeutic effect. Okay, so you basically starting with a biological hypothesis, and you need to demonstrate that hypothesis by having compounds to really activate or inhibit that enzyme and have its biological effect. Most of the time, biologists will go and they will find public information. It will be a chemical structure, some possible X-ray data, and they will start with working on that biological hypothesis, what I will call target hypothesis. And in order to slowly validate your in vitro assay and further down the line, validate your in vivo assay. Okay. So usually big pharma, they tend to prefer working on validated targets because of the price it costs basically to deliver. And also new targets, they are very interesting, but the probability of delivering is even lower. Okay. So you developed your biological assay, your in vitro assay. Then what you will do, you will have a screening compound collection. So this is basically up to like 1 million of compounds, commercial compounds that you're buying as a cassette, and you will be screening them in vitro and looking for actives. So this looks like all pretty good, but it's 1 million compounds, and you have someone in the lab that is going and testing those, even though they are robot formats. It's still expensive. There are issues with those screening cassettes. Most of the time you have false positive. It could be on the way you plate your assay. It could be also the compound itself. There could be compounds that are what we say promiscuity. that are promiscuous, so they will just pan assay and hit everything, and you will think that you have already a very good compound. Okay, so you have a lot of work to do to prove that you have an active, and then you will go and do a quality check on the compound, and you will find that it's actually some impurity that is giving you activity, and then all your cassette screen might not be very fruitful. So for this, you could have some computational chemists that will come and will do a library design. So it will be a more focused, if you want, screening compound collection, still coming from a commercial compound. But all these are very expensive. So this is where you have more and more research being done in terms of in silico design and the development of tool in terms of how I can do these without having to test so many compounds. Okay. So I'm going to talk just about 3 methods for early stage hit generation. The first one is called Blaze. Blaze was developed by the Cresset Group. So it is basically a web application, very easy to use. And what it does, it's a virtual computational screening of compounds, if you want. So it's still commercial compound that you can go and buy if you want, but instead of having to plate all of them, you're just going to do a screening with the tool. Okay, so what it does, Scott and Rob mentioned about what is the biological activity that you're seeing. It's about this relationship between a protein and a ligand. Why is it that they have this interaction? It's a lot due to the shape, but also a lot due to the electrostatic effect. So the way we are representing the chemical compound is not like you will draw a 2D structure. We are really thinking in terms of 3D. So this is what Scott has called the pose. It's the conformation. A compound in solution is going to adapt a multitude of conformation. You have the ones that are the happiest, that are the most likely to exist, but you still have a lot of them. Okay. And on the same side, you have this huge protein that is doing the same thing. Okay. So you will start with a hypothesis about that particular pose, that particular conformation of that ligand with that protein, and we will call this a bioactive conformation. Okay. So this gives you already the 3D shape, and in the same time, you will take into account the representation of your ligand, but based on the electrostatic fields. Okay, Scott mentioned about the positive and negative fields around, but remember you have a lot of van der Waals hydrophobic interaction that are also very key in terms of interacting with your protein. Okay, so this will be your hypothesis. You will have a bioactive conformation hypothesis, and in the same time, you will take into account electrostatic fields. So this could be coming from your initial tool compound that you find from the literature, let's say, but you want to find a lead that belongs to you. That is your reference compound, what we call a query molecule, and you can do that with or without a protein. Okay, so it's a method that you can do for both. And what you will do, you will screen your whole commercial cassettes and found compounds that are very similar, not necessarily as a 2D structure, but more as a shape and electrostatic field representation. Okay, so this is more or less the way the method is. Okay, and here I'm giving you an example. So this is the receptor, I think, yes, the receptor is called 11 beta HSD1. So this is an enzyme that catalyzes the reaction. You see the cursor? Yes. So this is an enzyme that catalyzes the reduction of the cortisol, the ketone here, into the alcohol cortisol. So cortisol in itself is inactive in the cell, but once it is as the alcohol, it starts having some effect in terms of a lot around the metabolism, but also in terms of inflammation response and etc. So you can think about one enzyme that does this, and then you can link inflammation with and metabolic reaction to potential disease. So think about oncology with the metabolism, think about inflammation and oncology, think about inflammation and CNS issues, Alzheimer, there are a lot of hypotheses around. Think about inflammation and autoimmunity, for example. All right, so same process as I mentioned. I'm not going to go over this too much now. So you will, this is the way it's being represented. So you see that it's supposed to be a 3D structure, and then the dots and balls are representing your field, orange and yellow for the hydrophobic van der Waals interaction. Okay. So, and then you will have a match that is being found on your database. So the Blaze database, I think it's over 10 million of compounds. These are unique compounds and these are commercial compounds. You can add more databases if you want, but it's a fee base, so you will have to pay more. Perfect. So for this process using Blaze, what we found 3 different compounds, and you can look in terms of the structure. You can consider them to be novel series, different chemical series. Okay, so this gives you now 3 different starting points to go ahead and test your biological hypothesis. Okay, and these ones were similar from a known compound for this particular target with competitors. And this one form the base of a patent for one of the customers of that Blaze. Okay, there are lots of things that you can do. So this was just a process, but adding to that, you can also do docking score, electrostatic complementarity scoring if you know more about your protein. Okay, so there are orthogonal methods that you can add. So this was the first method. Now we are moving into the second method. It's also a method developed by Cresset. It's called Spark. And instead of now looking at a whole molecule design, what you're looking at region of your initial ligands. So we call this bioisosteric replacement. Still using the electrostatic field points and the 3D structure pose of bioactive conformation as your guide, for your search, what you're doing, you're replacing a portion of that molecule. And it's quite a powerful bioisoster replacement tools. And you can see here is just, we are just looking at a replacement of a phenyl portion into here. It looks quite basic, a pyridine and a thiophene. Who wouldn't have thought of this? But I'll come into a more detail to that. Okay, so what I find is particularly powerful is the way you're looking at scaffold hopping. So you start with that tricycle compounds there on the left-hand side, and imagine about a disconnection where you're changing the core. This is now where it becomes very powerful. You can think about, if you bring your chemistry, organic chemistry hat, you can already think about potential reaction disconnection you can do. You can think about what reagent do I have in the chemical inventory in my lab, and think at quick ways of addressing your issues and making those on. So here, same idea. So here you're doing a bioisoster replacement, and there were 3 new scaffolds now that were identified. You can ask a question, why do I call these scaffold? Because if it was on one of the substituent outside, it's still the same platform, but there are analogs. Whereas here you're changing the core, and we call them different scaffolds. Okay, so these could form the basis of 3 different patterns, if you want. So the process, so initially for the fragments, you're just looking at a geometric replacement of a geometric search of that fragment. Is just a conformational search, if you want. And then once the computer has retrieved your software has retrieved all the different interesting fragments from that commercial database, it will build every single ligand fully, and then acquire the electrostatic field point, and then do the spark docking the spark scoring, if you want. Okay, and this is when you will go and do the analysis and prioritization and selection. What is interesting is about the database that are included in Spark. So you have, this is the one that is the most commonly used and very easy to search, the chemical database, the ChemMol database. These are commercial compounds and compounds that are known in the literature. Okay, so, and then you also have the zinc database. These are more like specially curated commercial compound as well, but you need to pay for this to be able to do that search. Okay, so once you have a whole molecule scoring, so this is the sort of value that you get between 0 to 1, and these 3 were the one that you had the highest scoring, and they were the one that was chosen to pursue. So the way you will, from one ligand, the way I will approach the use of Spark, I won't go and do one by one. I will do 3 spark searches simultaneously. And I will look at one where I'm replacing the core, bioisoster replacement there. It will give me new platforms. And then I will look very quickly at a way to quickly find alternative groups there. Okay. So that's, I think it's quite a powerful search I found. So here is just to show you what it looks like in terms of 3D, but also in terms of the way the electrostatic fields are represented. We did a search initially, it was just based on the conformation of this part, and then look at the effect between the green initial ligands to the end result ligands. Look at the effect in terms of the electrostatic field. Although we changed just this region of the ligand, you can see its domino effect into the other region as well. So this is why we do the whole calculations at the end. Okay. Any questions? So now moving on into a third possibility. So now it's a case where you're going to do a screen, but you're not going to do a full-blown ligand screen. You're going to look for fragments. Why do you want to look for fragments when you can look for already made-up ligands? So imagine you have a hit and your hit is 500 nanomolar. You will say that's pretty good for a new starter hit. But I'm sorry, if your hit is already 450 nanomolar, sorry. If your hit is already, let's say, 450 in terms of molecular weight, it's not a great starting point, isn't it? Because you will have to optimize that ligand to improve both in terms of activity, but also PK and etc. to have a better pharmacological effect. So because that is a bad starting point, what you will really favor, you will favor a smaller starting point in terms of molecular weight. And you will say, okay, I don't mind if I have a 1 micromolar activity. I know the molecular weight is only 250. I have room to optimize, and then you acquire knowledge, and it's becoming more rational design rather than a search food blast. Okay. So here I mentioned that the ligands might have suboptimal interaction with your protein. So it's just a schematic representation. Okay, and I imagine a ligand that had free interaction domain with the protein. Now on the other side, you have a receptor with one fragment, a fragment A, that has some activity. Activity may not be great, but actually it is small. It has already optimum interaction because I managed to measure that. Okay. Imagine you have another fragment. And imagine you know the structure of that protein, and you know that those two fragments, they bind at different region. So you're starting to have a better understanding in terms of what constitute the potential active site of your protein, the different pockets where you know ligands are interacting, and then you can think of experiment like growing into new region that you know have an effect in terms of pharmacology, but also in terms of joining those two and see if it's an additive effect. Okay, so you're understanding things more and in a more rational way. Okay, so you can do those two things in Spark, and it's quite powerful. The growing experiment and the joining experiment, and I've been very lucky to have many successes with those. Okay, so. Here I'm just going to show you an example. It's the P38 alpha protein. So don't think about the whole, try not to understand the full image. I'm not going to go into details. We will look at it more when we go into the hands-on practice tomorrow. So here what we knew that that fragment was binding, and we could see that binding pocket, there was a region where we can go into some and interact with some amino acid of that binding pocket. So you can think here, it would be a growing experiment, yes? So that's what we did. So we used Spark, and then the result, we brought them back into Flare, where we are, it's the software where we are looking at the protein structure with the ligands. Okay, and then based on those scoring, we went and had these ligands. I think this one was one of the top 5 ligand that was designed using Spark. And this is the competition. So Pfizer had a compound, and that was also binding to that P38 alpha protein, and have a look at it. So very similar, I will say, they bind in the same pocket, yes. They have very similar interaction with the amino acid on top. And then you can see this region, our ligand seems to be shorter and have more as an anchor compared to the Pfizer ligand. So this is just an illustrative example. This compound didn't go and made a drug. It was just to show the thought process and how it works. Okay. So I think, yes, that's it. So as a conclusion, I've shown you 3 different methods to help you to generate in a more rational way your starting point, if you want, on a project. The Blaze, with the Blaze computational screen, you get an increase in terms of hit rate with less compound to be screened, so more in terms of efficiency. And then there are different ways with Spark to innovate and open your IP space and do a hit expansion in a very quick way. Okay. So, and then we will be practicing tomorrow with Kevin for those that are coming. And that's it. Thank you for your attention.
Selma, so I know that Selma and I are between you and your lunch at this stage, right? So anybody got some questions from the audience? Young man here again.
Yeah, hi, that's fascinating. So I had a quick question. Based on shortening your list of possible hits, can you do the same with pharmacokinetic prediction? So like say, rather than just model ligand versus target, you could model ligand versus, you know, cytochrome B450 or whatever,
Yes.
it's going to break it down.
Yeah, off-target activities.
Right, and
Basically.
predict, well, you know, this is what's going to metabolize the ligand after I've made it.
Yes.
Will shorten my list even more.
So yes, so for example, if you're talking about first path metabolism, for example, you tend to look more, rather than this being, obviously there are some known pharmacophore for metabolic liability, but usually it's the overall physical properties of your compound. There are strategies where you can leverage your physical properties and see an effect in terms of metabolism. But I guess this may be the second part of your question. It could be, you can also look at off-target activity. But for this, instead of looking for an activity model, you will build a model of something you don't want, basically, and look at scoring. Yes? So you can do that for the selectivity. So if you know about the binding site, you can do some docking for this, or otherwise you can build models. And these are things that we do as well.
Is there any question online? No? Okay, so you can have the last question of the session.
So for all of these screens, you start with ligands, right? In theory, could you start without a ligand?
So in theory, can you start without a ligand? You will do, most likely you will do a compound screening, like a proper screen. But the way I will approach that, you start with a biological hypothesis. So how are you going to validate that hypothesis? You're going to search the literature and then find a tool compound that might not be the best, but will have shown some activity in your in vitro assay. Yes, and this is where I will start if you don't want to do the screen, and then I will go slowly on from this. Thank you.
Okay, so thank you, Selma, and thanks to Scott as well in that session. So we're just a little bit over time, but you've still got till 2:10, right? So you have an hour and 5 minutes. If I can do my calculations correctly for lunch, which I presume is in the same place where coffee was earlier on. So again, just for me, thank you to everybody, all the Cresset speakers and Cresset for coming over again. Thank you all for attending, and I'll see you in the afternoon. So thank you all. Bye.
He's going to put slide.